Hi everybody, welcome to Unit 8, the nervous system. We're going to talk about the central and peripheral nervous system, the general functions of a neuron, how electrolytes are used for nerve impulses, the difference between sensory interneuron and motor neurons. We're going to talk about reflexes, the parts of the brain, the parts of the spinal cord, and compare the sympathetic and parasympathetic nervous systems. So the nervous system is divided into two parts, the central and peripheral nervous system. The central nervous system is in gray in this diagram here, the brain and the spinal cord. Everything else, all the receptors and sensory detectors, nerves, are considered the peripheral nervous system, so everything in the rest of the body. The central nervous system, again, is your brain and spinal cord, and the rest of the nerves are the peripheral nervous system, and that's the one that really is we're more aware of, I guess, and also the one that we can repair if damaged compared to the central nervous system. Often one of the problems with the central nervous system is if there's damage, it doesn't repair or doesn't repair the same way or back to the original. The functions of the nervous system are to detect information with sensory receptors and send that information to the brain, which figures out what it means, that integrative or processing part, and then the brain decides to send a response through the motor neurons to an effector. Effectors are always muscles or glands. So we have all these sensory receptors all over the body, some of them outside, some of them inside. Probably the most common is what you learned when you were in grade school, for example, the sense of touch, taste, smell, hearing, and sight. So those are sensory receptors kind of on the outside or the periphery of the body that take information to the brain. But you also have sensory receptors inside. Inside your carotid arteries, it detects blood pressure. You have osmoreceptors that detect electrolyte balance and water balance in your brain. You have sensory receptors that detect your blood glucose levels in your pancreas, for example. Anyway, the sensory receptors, all different kinds, exist both on the periphery and on the inside of the body that's telling your brain all the time what's going on. It's kind of a quick little video describing these quick functions here. If you want to stop and check this out, go back to the original PowerPoint and check that video out. So for example, if you see an apple, your eyes pick that up, photoreceptors, that information goes to your brain. The brain says, I've seen what that is before, determines it's an apple, sends information out, not only so you recognize what the item is, but if you want to pick it up, it would send information to the muscles in your arm and hand, allowing you to grip and pick up that apple. So again, stimulus activates a receptor. Sensory neurons or nerves take the information to the brain. Motor neurons or nerves take information away from the brain to an effector. An effector is always a muscle or a gland. Alright, this is a picture of a bunch of neurons. Neurons are the structural and functional units of the nervous system. They are specialized to react to physical and chemical changes in their environment. The main black part of this is the cell body, and they have branches or fibers extending from the cell body called axons and dendrites. The axons send information away from the cell body, and dendrites receive information or input and send it towards the cell body. They secrete neurotransmitters, which are chemical messengers, into synaptic spaces between neurons, and then that triggers the next neuron to send another impulse or inhibits it. They have the ability to send impulses or to block the sending of impulses. It is electrical as it moves down the axon and chemical when the neurotransmitter is released. So sometimes neurons are called electrochemical signaling molecules. They are surrounded by neural glial cells. Neural glial cells function to support the neurons. Okay, so they might hold blood vessels, they might help clean up messes or protect them from pathogens, that sort of thing. A mass of neurons or a mass of axons make up what we call nerves. Okay, so that's the general definition of some of the parts of the nervous system. The functional unit of the nervous system is the neuron. Here's another picture. The neurons are colored yellow in this picture, and the neuroglial cells supporting them are in green. The average neuron has a cell body with extensions kind of sticking out of it. One of the extensions would be the axon. There's always one axon, which might branch at the end, but it is the long one where the impulse goes from the cell body towards the end of it, or away from the cell body. And then it may have one or more dendrites. Dendrites, at the end of the dendrites, that's where the sensory receptors are located. And when they're activated enough, when they hit threshold or the minimum amount of stimulus, they send that information towards the cell body. So axons send the impulse away. Dendrites are fibers which conduct impulses toward the cell body. Here's another picture of a neuron, and at the end of the axon terminals, there are synaptic knobs where the neurotransmitter is released. Axons that are, neurons that are in the peripheral nervous system, those axons are surrounded by what are called Schwann cells that lay down myelin around it. Those little white sausage-looking things there are Schwann cells that have wrapped layers of myelin, which is kind of like insulation, around the axon fiber. Okay, so Schwann cells lay down myelin. The cell membrane of a Schwann cell is called the neurolemma, and the presence of that makes it so if that axon was damaged or cut, it could be repaired. Okay, and Schwann cells are only in the peripheral nervous system, so there's no repairing axons or neurons in the central nervous system like there is in the peripheral nervous system because of the presence of those Schwann cells. In between each Schwann cell, you can see a little bit of the axon there in yellow. Those places or spaces between Schwann cells are called nodes of Ranvier. It's a French scientist who figured that out. So those little nodes are the spaces between the Schwann cells along the axon of a neuron. So the nerve impulse is initiated once the dendrites are activated enough. So if this is a touch receptor, you'd have to touch it hard enough or with enough pressure so there's a minimum amount of stimulus required. That signal goes towards the cell body, and once you have enough of the signal, the axon propagates down, the impulse propagates down the axon away from the cell body to the synaptic knobs where a neurotransmitter is released. So basically a nerve impulse is initiated when there's enough of an incoming signal to trigger a response. It causes ions, charged atoms, remember that from Unit 2, to start moving across the axon membrane, carried all the way down to the end where a neurotransmitter is released. And that's what we're going to talk about in the next few slides, is how that nerve impulse works. So basically, a neuron at rest has more sodium on the outside than it does on the inside, which makes a charge difference occur. There's more positive ions on one side than there is on the other side. So the membrane is said to have potential, because potentially those charges could change sides. So at rest, there's membrane potential, or the neuron is polarized. Okay, and you spend a lot of calories every day maintaining this potential, just in case you touch something hot, or you step on something painful, or something is, suddenly thrown at you. You can blink and turn your head away, almost before you stop. Before you stop and think. Those are reactions that you can do because your neurons are ready to send an impulse. Anyway, so this potential exists all the time at rest, waiting for a nerve impulse to need to be sent. So if there's a movement of charges because of some sensory input, that's called an impulse. Okay, and a current is going to, when charges start to move, that's basically a current. So if the charges move, you get a current, which in this case is an impulse. So in this picture right here, once you get enough impulse or sensory information, the impulse starts to go because those charges, those ions, start to move across the membrane. And you have a current now because you have electrical charges moving. So at rest, a normal neuron is electrically charged or polarized. It has potential. The inside is more negative than the outside, or the outside is more positive, whichever way you want to say it. You can actually measure that. At rest, there's a 70 millivolt difference between the inside and the outside of the cell. Okay, so at any time when you get enough stimulus, what actually happens first is the sodium on the outside, which is a positively charged ion, rushes in because channels open up. So that positive on the outside becomes more positive on the inside. You can see that in this slide here. So the first ion to move when you get a stimulus is sodium rushes in. And you can see the little green balls on the right side is rushing into the cell. Now it makes the inside more positive. This is referred to as depolarization because you were polarized to begin with, but now you're depolarized because the amount of positivity changes, more positive on the outside changes to more positive on the inside. To counteract that, immediately potassium channels open up and potassium rushes out. Potassium is also positively charged, and so now you have the positive going back to the outside. This is called repolarization because you went back to what you were at rest. The problem, of course, is sodium's on the inside and potassium's on the outside, and it's supposed to be the other way around. But in the meantime, this goes all the way down the axon. So if you remember this picture here, you go from 2 to 3 to 4 to 5 to 6 here. The sodium rushes in, potassium out, sodium in, potassium out, sodium in, potassium out, all the way down to the end. And at the end of the axon is the synaptic knob, and there's neurotransmitters and vesicles waiting there, and that change in charge causes an influx of calcium, which causes the vesicles to merge with the cell membrane and release its contents. So those little tiny dots there into the synaptic space are neurotransmitter. That neurotransmitter will bind receptors on the next neuron and either block or initiate another nerve impulse in that neuron. Okay, so step 3 is the release of the neurotransmitters. In the meantime, all the way down back at the beginning of the axon, you have a pump called the sodium-potassium ATPase pump, putting the sodium back out and the potassium back in. It pumps 3 sodiums out for every 2 potassiums that go in, and what that does is restore membrane potential. Okay, you get back to normal. But you have to burn an ATP every time you do this, and these pumps work continuously. That's why, remember in the last unit, when we were talking about cellular respiration and BMR, I think that was the end of unit 3A, 17% of your energy requirements is in your brain, and it's mostly that little pump, pumping sodium back out and potassium back in so that your neuron is ready to send an impulse again right away. You can see that all of this happened kind of in action in this little YouTube video, so you should check that out. Go back to the original PowerPoint and watch the Action Potential YouTube because it really is kind of helpful. But in general, sodium rushes in, causing depolarization. Potassium rushes out, causing repolarization. The neurotransmitters are released at the end. And in the meantime, those pumps return sodium and potassium to the correct place. Sodium going back out and potassium going back into the cell. You can actually measure this. Okay, this graph is What's it measuring is the y-axis. Remember, the dependent variable is voltage, and you're comparing across time. So you get a stimulus, and once you get enough to hit threshold, depolarization, sodium rushing in, repolarization, potassium rushing out. It takes a little bit, and then the pumps basically restore a resting state by pumping 3 sodiums out and 2 potassiums back in. Now, the minimum amount of stimulus you need is called the threshold potential, or the threshold level. You have to have enough to initiate the contraction, but once you do, it's all or none. If you hit threshold 4 times, it doesn't matter. The depolarization only is going to go up to a certain peak. It's an all or none response. So more stimulus just means more impulses. It doesn't mean the impulse itself is bigger or stronger. And then that little dip there from repolarization back to resting state, that's called the refractory period. That's the time it takes for a neuron to recover so another impulse can be sent. It's pretty short. It's only 1/2500th of a second, but you do need to recover in order for another impulse to go, because the sodium and potassium are on the wrong side of the membrane after repolarization. But basically, if you touch something hot, you will jerk away. It doesn't take very long. And if you are a moron and touch the same hot thing, you're going to jerk away, because the refractory period is so short, and your neurons get back ready because they want to protect you, even if you're a moron, and touch the same hot thing twice. So it's kind of cool how it works, and it's amazing because most of the time, it works every time, and we don't even have to stop and think about it. But you do need to have sodium and potassium around, which is why it's so important to balance your electrolytes, which is what in the medical field you might worry about if patients come in with too much sodium or too little sodium or too much potassium or too little potassium. You start not to be able to send nerve impulses, and even thinking becomes very difficult to do. Okay, so how fast does this happen? In your brain, the axons are not myelinated. That's the top picture. So as you watch that little impulse go down, you can see how it's pretty slow. Okay, but there's not very far to go from one side of your brain to the other. So it's, happens in split seconds. But the signal in myelinated neurons, the ones that have Schwann cells, this impulse jumps from node to node to node, and that's the bottom one. And you can see it's so fast it's almost hard to watch, because it's a split second on the screen and then it's done. Okay, so nerve impulse conduction is actually faster in the peripheral nervous system, in your arms and legs, than it is within your brain. All right, what about those neurotransmitters? There are lots of neurotransmitters. They keep finding more. Some of the most common ones are listed here: acetylcholine, dopamine, norepinephrine, serotonin, and GABA, or gamma-aminobutyric acid. And they know that when people have more or less of these, it can cause some various diseases, but they're not. It's really hard to measure the levels in somebody's brain, okay, because how are you going to get to it? These neurotransmitters aren't necessarily going to show up in the blood. You'd have to stick a needle in the middle of your brain and suck fluid out. It's not even possible. So that's why often with anti-anxiety medication or depression medication, some of them are used or are meant to work by increasing the levels of certain neurotransmitters. They have to play around with the dose to find out what works best for each patient, which can be frustrating for the patient, I would imagine. But they know, for example, norepinephrine tends to be lower in people who are depressed and tends to be increased in people who have schizophrenia, manic depressive disorders, or anxiety. So, do we give it extra to people? Do we prevent breakdown? There's all different kinds of ways to treat mental illnesses. Okay, so that concludes kind of part 1 of the nervous system. This is a good time to stop and come back later if You can't do this all at one time. The second section is about the parts of the brain and spinal cord, and then there's a third section about the parasympathetic and sympathetic nervous system. So I'm going to go into part 2, but stop if you need to, and come back at the 20-minute mark. So, the first part of the brain that we're going to talk about is the highest part of the brain, called the cerebrum. You have a right and a left hemisphere, and they are connected by the corpus callosum, which is this white streak right here. And the cerebrum is everything above that. Okay? Can you tell which side of the brain this is and what kind of cut it was to create it? Hopefully you know this is a sagittal cut, and this would be the left side of the brain. Okay, besides the corpus callosum, a couple of other things you should know. The brain is bumpy or grooved. The main separations of the cerebral lobes are called fissures, but the little individual indentations are called sulci, or sulcus for singular. And it turns out the bumpier the brain, the smarter you are. People who have smooth brains are generally have mental impairments and other things. So I guess it's kind of an insult to say you must have a very smooth brain. So here's the different lobes of the cerebrum. In red, you have the frontal lobe. In blue, the parietal lobe. Green, the occipital lobe. And yellow, or goldish, orangish yellow, is the temporal lobe. All of it is the higher order thinking, the figuring out, and personality of you, but there are functions that tend to go with each one of those, and in this diagram it even talks about parietal lobe, and a lot of the senses, touch and language go on there. Occipital lobe is highly associated with your vision and interpreting visual images. The temporal lobe, I remember this because tempo, it's your music, hearing, balance. And the frontal lobe is more your personality, concentration, planning, problem solving, that type of thing. Some other cerebral structures include the hippocampus, amygdala, and basal nuclei. The hippocampus and amygdala are basically involved in memory, particularly long-term memory formation, and how you respond to things emotionally. You know, in people who have PTSD, their amygdala is actually smaller. The basal ganglia, or basal nuclei, is a place in the cerebrum where motor relay center occurs, or where motor impulses are sent there first, and then the basal nuclei or ganglia direct it to the right body part. They know that the main neurotransmitter in the basal ganglia is dopamine, and when the cells stop making enough dopamine, that's Parkinson's disease. It's a disease that tends to affect people as they get older, but Parkinson's and Alzheimer's are probably the two most common older mental disorders that people get or have. And they've done all kinds of things to try to treat Parkinson's, and one of them is give people dopamine, and that seems to work for a while, but they have even transplanted cells from pig brains from their basal ganglia to try to treat Parkinson's disease. Very interesting. Dopamine actually is an inhibitory neurotransmitter, and it inhibits the impulses going to all the motor neurons to all these muscles. And so without dopamine, because there's no inhibition, those muscles contract, and that's why people who have Parkinson's disease have tremors. The middle part of your brain is called the diencephalon. It includes the thalamus, the hypothalamus, the infundibulum, the pituitary gland, and the pineal gland. So if you look at this picture where it says thalamus, the thalamus is kind of right in the center of your brain, and it is the relay center for sensory information. So when you see something or smell something, the information goes to the thalamus, and the thalamus says, oh, here's something visual, I'm going to send you to the occipital lobe to figure out what it is. Oh, you are a smell, you need to go to this part of the parietal lobe. Or you are something you're hearing, I'm going to send you to the temporal lobe. Underneath the thalamus, or kind of forward in front of it, this brain, the eyeballs would be on the right side of the screen, so forward is, or facing forward is to the right there. So down and a little bit anterior is the hypothalamus. The hypothalamus is considered, to me, the master gland. It controls hormone release, it detects osmolarity, body temperature. It is, there's little reward centers in the hypothalamus when you eat, drink, or have sex because you're living or reproducing. It controls everything released from the pituitary gland, which you can see that labeled there hanging down. The connection between the hypothalamus and the pituitary gland, that little stalk right there, that's the infundibulum. So the infundibulum connects the hypothalamus to the pituitary gland. The pituitary gland secretes all kinds of hormones and pretty much regulates almost all of them in the body. The hypothalamus does by telling the pituitary release this or don't release this at different times. The pineal gland also secretes a hormone, melatonin. We'll talk about that in the next unit, but melatonin is released more at night than during the day, and the up and down release of melatonin is thought to be our way of keeping time. Okay, so somehow the body knows that you're 12 or you're 50, and menopause hits if you're a woman, and at 12 puberty hits if you're a boy, for example. So the diencephalon does a lot of the keeping you alive stuff, but also regulating homeostasis. The lowest part of your brain is known as the brainstem. It has 3 parts. The upper part here is the midbrain. Then there's a part that looks a little bit like a pregnant tummy. That's the pons. And then the part underneath that is the medulla oblongata, which connects you to the spinal cord, which is the lower yellow part here. The midbrain, or the brainstem, connects the brain to the spinal cord, and it has a lot of lower level functions. Sometimes it's called the reptile brain because that's pretty much all reptiles have. The midbrain is the reflex center for sight, hearing, helps you maintain posture. So even a baby will turn its head towards sound or light, and that is all the midbrain. The pons basically regulates breathing rate, the rate and the depth of your breathing, and the medulla oblongata also plays a role in that, in breathing, but also your heart rate. All the nerves leaving the brain goes through the medulla oblongata, and it connects the brain to the spinal cord. Okay, so that is the brainstem. Hanging off the bottom of the brain, back, kind of behind the brainstem, is the cerebellum. Okay, it looks a little bit like a bush there in this picture, and its main function is balance or posture. Communication between the cerebellum and the rest of the brain is via these things called peduncles, which you guys don't have to remember, but it's kind of a cool word. But basically, it tells the cerebrum where your body is, and the cerebrum figures that out and knows where the body wants to be. So if you're leaning towards the right, the brain can tell whatever muscles are needed to move you back left. So the peduncles, the cerebellum, is telling the brain where you're at, but also where you need to be, and the brain figures out how to fix that. Here's a sagittal section of a human cadaver brain, and the parts are labeled here so you can see the thalamus, the hypothalamus, the midbrain pons, the medulla oblongata. You can see the corpus callosum there. Connecting up above would be the cerebrum, and this section right here you can see part of the cerebellum. Kind of a cool brain. Somebody did a really nice job dissecting that one. Within the brain, there are also some spaces, or sacs, I guess, cavities, filled with cerebrospinal fluid. Cerebrospinal fluid nourishes the brain, it cushions the brain. If you get hit really hard, concussions can be pretty dangerous, but they would be worse if you didn't have this fluid helping the brain not, rattle back and forth inside your skull. Surrounding the brains are protective membranes called meninges. Again, it functions to kind of hold the brain to the skull. Between the arachnoid and the pia mater, these are the three layers. That's where the cerebrospinal fluid is. So that cushions, it nourishes the brain. So these membranes are both protective and nourishing. The dura mater is the one right underneath the skull. And then the arachnoid mater is underneath that. It looks like a spider web, hence the name. And the pia mater touches the brain itself, goes down into each one of those little grooves. And the cerebrospinal fluid is between those last two. It also extends down into the spinal cord, which is shown here. That little tiny circle in the middle of the spinal cord, it's called the central canal, and cerebrospinal fluid is in there too. So when you get a spinal tap, they go between two vertebrae and they put a needle all the way into that little circle there and suck out a little fluid to analyze it. So this is looking down at a thoracic vertebra, and the person, if you're looking at this, the top of the picture is the back of the person. So when you feel the bumps along somebody's vertebra, you're feeling that point right here of the vertebra sticking up. Okay, so this is the main body of the vertebra, is in front of that are your guts. Okay, so that's the orientation. Your spinal cord is within the vertebral foramen, or that hole, and they just drew that picture in there to show you. The next picture is just the spinal cord itself, so all the bone is gone from here. This part that looks like a butterfly is gray matter. It's not myelinated, and damage to that is generally permanent. Around it is white matter. Okay, so myelin is around the outside. The dorsal root is bringing information, sensory nerve information, to the spinal cord, which goes up to the brain to be interpreted. And information going out to the effector goes out via the ventral horns, or the efferent signals. Afferent and sensory are the same thing and used interchangeably. So an afferent nerve is a sensory nerve. It's bringing information to the spinal cord up to the brain. Efferent signals go to muscles or glands, effectors, and are the same as motor neurons or motor nerves. Alright, so there's a little bit about the spinal cord, and you have a picture of this in your lab book you can look at as well. You have two kinds of nerves, cranial nerves and spinal nerves. The cranial nerves branch directly off the brain. There are 12 of them, and for some reason they were named or numbered 1 through 12 using Roman numerals. Cranial nerve number 1 is the olfactory nerve, so it basically carries information from your nose to your brain. Some of them are sensory, some of them are motor, some of them are both. You can learn the function of each one if you're interested. We have a lab that tests each one. Doctors will sometimes use those same tests to determine if your cranial nerves are working. Okay. Number 1 is olfactory, number 2 is optic. The oculomotor, trochlear, abducens, which is number 3, 4, and 6, all are involved in moving your eye. The trigeminal nerve has a whole bunch of jobs. It's both sensory and nervous, facial expression, eye movement, chewing, all kinds of things. The facial nerve does a lot of facial expressions and sensory, or the touch when somebody touches your face. Number 8 is the vestibulocochlear or auditory nerve, so that's hearing. Number 9 and number 12 have to do with your tongue. Glossal means tongue. Taste, mixing of food, salivary secretion. The The vagal nerve is one that goes more than just your face. There's nerve impulses from some of your internal organs, including your heart and diaphragm, I believe. And it's such a busy nerve, the accessory nerve helps it. Number 10 and 11 kind of work together. So that's a general overview of the different cranial nerves. And they all have names. Spinal nerves do not have names. They're just numbered by the vertebra. So cervical vertebra, you have 7. You have C1, C2, C3, C4, etc. And thoracic, you have 12, so T1, T2, T3, etc. And they are both sensory and motor. The sensory nerves send information up to the brain, and the motor neurons send information from the brain to an effector. They innervate different regions of the body, and these regions that they innervate are called dermatomes. So here's a picture. T1 would control everything in that region. T7, that region. Lumbar vertebra number 4 is down the inside of your leg, for example, 3 and 4. Okay, so you can kind of see that if you had a back injury down at T4, everything below that is not going to work because you've cut the spinal cord or broke your back at that point. If the spinal cord is severed or damaged, everything below that's not going to work. Right? So dermatomes are the region that a spinal nerve controls or innervates. Okay, so that is pretty much the anatomy and kind of the function of the parts of the brain and the spinal cord. The last few slides here are going to talk about the physiology of the nervous system. If you need to take a break, now is a good time while you synthesize all this information, because there's a lot in the nervous system, if you couldn't tell that already. Alright, so part 3, physiology. Again, we've talked about the central nervous system, the brain and spinal cord, the peripheral nervous system, the cranial nerves, spinal nerves, and how it takes information from sensory receptors to the brain and information from the brain to the effectors, which are always muscles or glands. The peripheral nervous system can be further divided into the somatic nervous system, which is basically your muscles, your voluntary skeletal muscles that we talked about in Unit 7, and the autonomic nervous system, which I like to think of as the automatic stuff. So this is controlling things like your breathing rate, your heart rate, whether your pupils are dilated or not, whether your kidneys are fully going crazy with filtration or slowing down, the digestive system, whether food is being digested or it's slowed down. This is controlled, all of these things, by the autonomic nervous system. The autonomic nervous system basically can be split or divided into the sympathetic and parasympathetic. And that's what these last few slides are going to talk about, comparing the sympathetic to the parasympathetic. We've pretty much discussed the somatic nervous system with muscle contraction, and that was back in Unit 7. So we're going to do sympathetic and parasympathetic here. So the spinal cord can do some of the body's reactions without even consulting the brain. Generally, these are called spinal reflexes or reflex arcs, and there's very few neurons involved. And this is something a doctor would test even if you're unconscious. If your spinal cord's working, your spinal reflexes should work. And it's a way to determine the difference between brain dead and not, because spinal reflexes will sometimes work, but brain function is not. So they can kind of determine how far up the damage is by checking your spinal reflexes. Reflexes are defined as automatic, subconscious, unlearned responses. You did not learn how to sneeze. You did not learn how to swallow. You were born with these reflexes. The one the doctor tests most often is the knee-jerk reaction, where he or she will tap with a little mallet on the patellar tendon, and if your leg kicks out, your knee-jerk reaction is working. Okay, so automatic, unlearned, subconscious responses. The parts of a reflex arc include some sort of receptor, a sensory neuron that takes information to the spinal cord. There may or may not be an inner neuron in the spinal cord, but there's always a motor neuron out to an effector. So the 5 parts are in red here: a receptor, a sensory neuron, an interneuron, a motor neuron, and an effector. And again, you touch something hot, you will jerk it away. Nobody had to teach you to do that. Even a baby will do that. Okay? Now, certainly some information eventually gets up to the brain and you realize, I touched something hot, it was a pot, I shouldn't do that, there's fire there, why did I do that, I really need to grab a mitt or whatever. Certainly the brain is involved, but the initial response of jerking away, that's the reflex part. Again, you've probably had this done to you in a doctor's office, the knee-jerk reaction. The receptor is in the knee. There's a proprioreceptor there. Sensory neuron takes information to the spinal cord. That's in blue here. There's an interneuron in green in the picture in the spinal cord. A motor neuron carries information back to the hamstring muscles, causes it to contract, and your knee or your lower leg moves. Okay, the quads and the hamstrings are told to contract or relax. The effector is always a muscle or a gland. Okay. The peripheral nervous system, again, is divided into the somatic and the autonomic. And again, we're going to talk mostly about the sympathetic and parasympathetic because we've already done muscle contraction. So what is the autonomic nervous system controlling? Things like your pupil dilation or constriction, your breathing rate, your body temperature, whether you're sweating, whether you're making urine, whether your blood pressure is high or low. It can up your metabolism or slow it. Basically, it's all the things that you're not thinking about, right? You don't have to worry about this stuff. My breathing rate is just kind of whatever it needs to be. Thank God we don't have to listen to all this stuff or figure it all out because we have things to do, right? Drive to school, go to work, study. Okay, we don't have to think, oh, today my heart rate needs to be this because I'm going to be doing yada, yada, yada. You don't. The autonomic nervous system will do this. The parasympathetic nervous system is what is in charge most of the time. Both of them are active or alive or working, but the parasympathetic is the dominant one during a normal day in the life of me, of whatever you're doing. During normal resting conditions, your digestive system is working, your kidneys are working, your heart's beating at a normal, regular pace, etc. It is the rest and digest state. Okay? The neurotransmitter that's released at all the effectors is ACH. They're called cholinergic receptors, and they bind acetylcholine. When something scares you or stress occurs, the sympathetic nervous system becomes activated, and it responds to emergencies. It is not meant to be turned on all the time. Okay? And people who have that happen can have some damage go on because we can't survive all the time in flight or fight mode, because that's what this one is. The neurotransmitter released is norepinephrine or epinephrine, depending on what you want to call it, and it makes you heartbeat faster and your eyes dilate and your breathing rate increases, and it slows down digestion and kidney filtration because more blood is going to be sent to your arms and legs so you can run or fight or deal with whatever the stressor is. And hopefully this is just a few minutes, because in general the parasympathetic nervous system is the one that is active and keeps you, at homeostasis and boring and level and normal, which is really what you want to be. But the sympathetic nervous system is activated to help you deal with the lion that's chasing you, or the deer that just ran out in front of your car, or when your kid falls and you didn't catch him. What are you going to do to help, that kid recover or whatever? The key point here is both the parasympathetic and sympathetic have the same effectors. Okay? They have the same things. Both of them make the heart do something. But generally the actions are opposite. So in this picture, for example, you can see here's the lungs on the left side and the lungs on the right. In the parasympathetic nervous system, breathing is normal, so your bronchial tubes are constricted down to a normal diameter. But if there's stress, it dilates those bronchial tubes, makes them bigger so you can get more air in, more oxygen to deal with the stress. The heart slows down in the parasympathetic activation, but when the sympathetic is active, it speeds it up. Okay, fight or flight. Note the effectors again are the same, the liver, blood vessels, the gut, okay, but the action generally is opposite. Over here, this neurotransmitter released at the effector is acetylcholine. Over here, it's epinephrine or norepinephrine. You can check this out in more detail in this video if you want to watch the comparison between the two and what happens. Again, go back to the original PowerPoint and check out these links. They're kind of cool. Okay, and it really explains the difference between the two. Last but not least, if a lot of things can go wrong with the nervous system, and we don't know very much about it. Even though, we've learned gobs of information, we still don't have a good understanding of how the brain works or how to measure the levels of different chemicals and hormones that might be there at any given time. If the person can't make a neurotransmitter or can't make the right amount, what might happen? And we don't always know. But nerve impulses aren't going to be sent in a normal fashion if the neurotransmitters aren't there. And how that affects the person might be different from one person to the next. But even worse, how do we know what the problem is and which neurotransmitter is screwed up and how can we fix it? We also know that electrolyte imbalance can really affect brains. Not only will a lack of glucose make you not be able to think, which is why you should have breakfast before you come to class, or lunch, depending, but what about sodium and potassium? Okay, if somebody's sodium and potassium levels were screwed up, you can imagine it might mess up nerve impulse conduction, because sodium, potassium, and calcium is so important in neuron impulse conduction. If caffeine, which mimics epinephrine or norepinephrine, was consumed, what do you suppose happens? Well, it makes your heart rate go up. It makes your liver produce glucose. It makes you more alert. Okay, just like the hormone does, norepinephrine. So we like that feeling. Some of us really like caffeine. We're addicted to it, right? Because it helps increase your blood sugar and makes you feel more awake. Lots of interesting things with the nervous system. I hope you have enjoyed this one. We are moving right through. We're about half done with the class right now if you've stuck with it thus far. Anyway, don't forget to check out the reading assignment for Unit 8, the lab manual for the various labs. There's lots of cool websites about the nervous system. Don't be afraid to Google some. And you, as always, email me if you have any questions.